And our next speaker is Mitch Tuinstra. He is at Purdue University, and he is actually the Wickersham Chair of Excellence in Agricultural Research, and he is Professor of Plant Breeding and Genetics. And his interest is in stress resilience of plants, increasing stress resilience, and also doing a lot of high throughput phenotyping in-house and in fields, I think. But today he will present to us integrated root and shoot phenotyping of crop plants in controlled environments. And Mitch, we are very interested and looking forward to your talk.
Thank you, Thomas. You can see my screen?
Yes, it's fine. Thanks.
So for the last 10 years, first off, thank you for the introduction. It's I'm sitting here. I'm actually at another meeting in the western part of the U.S. So I got up really early to get on the workshop so I could see the work that everyone else is doing. It's been really nice, a good day for seeing the breadth of work that's happening in Europe, especially. So thank you very much for the invitation. So for Purdue's been investing a lot of resources in high throughput phenotyping for the last 10 years or so. And one of the things that we've been very cognizant about in phenotyping relates a little bit to the conversation that we had with James around phenotyping at different scales. Are we phenotyping at a tissue scale, at a leaf scale, at a plant scale, at a plot scale, at a field scale, at a watershed scale? And not that any one way is better than any other way, but the fact that each of these different approaches provides different kinds of information into plant biology. And so we've been slowly developing a suite of different platforms representing these different kinds of data sets. Today, I'm just going to be talking to you. I'm going to first just introduce really quickly some work that we've been doing in establishing a field-based phenotyping platform, the pros and cons of that system. And then I'm going to shift over to talking about controlled environment phenotyping as an approach to dealing with some of the constraints and limitations of field-based phenotyping. So at Purdue, our field-based phenotyping platform is coordinated at the Indiana Corn and Soybean Innovation Center. The Innovation Center is a fully integrated field phenotyping facility that accommodates plant and seed analyses with proximal and remote sensing capabilities. This is a 25,000 square foot research facility at a 1,000 hectare farm, just 5 miles from campus. And so this is the workhorse of the sort of agronomic field sites for researchers at Purdue. There's over 60 faculty members from, I think, 6 different departments that do work at the agronomy farm. Is what this facility is known. Over the last few years, we've been working with faculty in engineering and the Polytechnic and in science, developing ground-based systems. That system that you see on the left there, that's the Pheno rover. It's a converted spray tractor that normally would be used to drive over the crop and spray the crop. But if you look at the boom on the Pheno rover and you look across the top of the boom on the front, we actually have 11 different station mounts there that can be used to mount different kinds of sensors. And in our sort of workhorse mode is we were using the Pheno rover to collect very high resolution RGB video, hyperspectral, and occasionally thermal data and LiDAR data actually. And so we have our ground-based systems. This is one iteration of our ground-based system. Our current workhorse is it's a UAV-based system. Our UAV-based systems, we've been really careful about multi-sensor integrations and calibrations. And so we have a multi-sensor, a series of multi-sensor environments. The most common system would be a LiDAR, hyperspectral Wiener, RGB system payload that essentially is used as the workhorse to acquire geometric, spectral, and three-dimensional data of crop plants growing in field environments. And we've developed the hardware, we've developed the integrated sensor solutions, we've developed software that can be used to process the data. And we've then transitioned these experimental platforms into a UAV-based plant phenotyping service. And so the Institute for Plant Science, one of the challenges that we realized early on is that many groups don't have the engineering support, or they don't have the science support, or they don't have the engineers don't have the agronomic crop science support to do these experiments. And so we developed this phenotyping service through the Institute of Plant Science. And this is really a system that was designed to democratize high throughput phenotyping with very high-end data acquisition and processing support for agronomists, our plant scientists across the College of Agriculture and actually across the faculty of the entire university campus. This system is in extremely high demand. In 2021, we flew hundreds of missions at the agronomy farm, and we actually also fly the Purdue Ag Centers around the state of Kansas. We have a number of Purdue Ag Centers within 1 to 2 hour drive of campus. And the system was essentially in the air almost every day last summer. One of the reasons why we do lots of field phenotyping, and I'm a plant breeder. I really value field-based phenotyping and trying to understand how plants are going to grow and develop. But one of the big challenges is if you're trying to do careful analysis of some biological phenomena that is of interest to you, as James just described recently in this last talk, it's hard to control. Well, you can't control all of the environmental factors that are going to sometimes confuse, sometimes cause variability and diminish the amount of genetic signal that we might be looking for. And so field-based phenotyping certainly has its place and it's important for us, but one of the very important aspects of controlled environment phenotyping is actually being able to carefully apply either environmental treatments or different kinds of other factors, management treatments to plants to better understand how are they interacting with regards to phenotypic expression. And so our controlled environment phenotyping facility is actually located on campus. It's located in the Ag Alumni Seed Plant Phenotyping Facility. It actually consists of 2 large conviron growth chambers with fully automated conveyor-driven plant production systems. And so these, the 2 conveyors, or the 2 growth chambers can accommodate up to 600 plants at a time with plants up to 4 meters in height. It was designed to accommodate full-grown hybrid maize from a phenotyping perspective. Since these are growth chambers, we have very good control of air temperature, relative humidity, and radiation PAR. We just recently retrofitted these systems for CO2 enrichment. So now we have growth chambers where we can have different types of atmospheric conditions with regards to CO2 concentration. And the system is on an entirely automated format where watering and fertigation can be defined from the plant manager. So that is our plant production system. And adjacent to the plant production system, we have a series of machine vision systems that enable our high throughput phenotyping applications from the standpoint of non-destructive phenotypic trait assessment. We have an RGB system where we acquire top view and side view data. We can collect data on approximately 120 plants per hour. We have a hyperspectral imaging tower where we can acquire top view and side view data on about 70 plants per hour. And we have an X-ray CT system that allows for root imaging at about 4 plants per hour. If you look at our RGB imaging platform, as I said, it's a top view, side view system. If you look at these pictures, you'll see a surprising array of different kinds of plants. We're looking at leafy vegetables, basil, kale, wheat, barley, sorghum, corn. We've looked at lots of different plant species. And so we're trying to be flexible with our platform in order to support the broader plant science community at Purdue University. And we're able to use the RGB imaging system to collect geometric, color, and predicted plant biomass traits for every plant that moves through the system. Our hyperspectral imaging tower, also side view and top view scanners. In this case, we're using both Wiener and SWIR, so roughly 400 to 1,000 nanometers in the Wiener and 1,000 to 2,500 nanometers in the SWIR. And this system, in terms of top view and side view, again, it allows us to collect an array of traits, some of them spectral indices, for example, but also we're developing algorithms that are trained to predict physiological traits and compositional traits as well. And lastly, we have the X-ray CT system. All 3 of these systems are integrated into the automation of the platform. The X-ray CT system has its own little dogleg because it takes considerably longer time to queue up plants and run them through the system. So you can't just drive everything through the system on a given day at 4 plants per hour, it would take a while to run through the entire growth chamber. But the X-ray CT system is being used to create 3D images of plant root systems. And the beauty of this particular system is that we can look at both shoot traits and root traits on the same plant on the same day and look at the relationship between these different plant traits and plant systems. This is a system that was actually, we co-developed with Fraunhofer. And so we actually have a spatial resolution of about between 75 and 200 microns. I was thinking about how I would go from here. How could I explain to you a little bit about the kinds of work that's happening at Purdue? I could describe all the calibration studies, for example, or I could describe my own research. But instead, what I decided to do was just to highlight a few case studies of some of the faculty members that I work with, just so you would get a sense of the breadth of applications that are possible across in our particular system. So for example, with the RGB system, we work closely with the wheat breeder, Mohsen Mohammadi. He's in the Department of Agronomy. His interest was in using the RGB imaging platform to develop machine vision and automated phenotyping for plant biomass, as well as staging of plants and looking at changes in plant growth in response to environmental stresses. He did an experiment. His calibration study was looking at a couple of different varieties of wheat. They were grown in the growth chamber. They were pulled out on the automated conveyance system. All of the plants were imaged weekly, and they were also ground truth so that he could relate his remote sensing RGB data with his biomass and leaf area data and developing predictive algorithms for variation in plant performance. And not surprisingly, this is something that one would expect to work quite well, and it worked quite well for him. And he's using this now routinely to calculate relative growth rate of biomass. Another one of our young junior faculty members, Professor Diane Wang, works on rice. And so Diane, or Dr. Wang, and her student were looking at plant nutrient analysis of rice. And so she took 2 different rice varieties, 2 different treatments for nitrogen, put them into a 12-week growth cycle, and demonstrated that she could collect hyperspectral data that could be processed using machine learning approaches to compute nitrogen content. Now that she's proven out that probably one of the easier traits to demonstrate a functional relationship, she's now looking at all of the other plant nutrients in terms of trying to use remote sensing to define and characterize variations in plant nutrient status, either defined by genetic effects or environmental effects. Professor Hoagland is in horticulture, and Professor Hoagland and her student Maria Rojas were interested in horticultural crops. And so the goal of her project was to use hyperspectral imaging to detect toxic metal stresses in basil and in kale. And so she did a study where she looked at basil and kale that were treated with various treatments of cadmium, 0, 5, and 10 parts per million. And she was able to demonstrate that she could use hyperspectral imaging to differentiate between the treated and untreated plants. And she's now developing predictive algorithms that actually describe the relative concentrations of these heavy metals in these green leafy vegetables. And her hope then is that this will be a system that can be used in environments that are prone to toxic concentrations of these heavy metals and can be used as an early detection system. The 2 pictured here, these are 2 postdocs. Valerian works with Anjali Iyer-Pascuzzi in botany and plant pathology. Monica Herrera-Huerta was a postdoc working in the Institute of Plant Sciences with Dr. Young. And these 2 have different types of backgrounds. One is more engineering and the other is more plant science related. And what they've been doing for the last few years is they're developing a data processing pipeline for our X-ray CT system. So as I said, this is a system that we installed in collaboration with Fraunhofer, and we started off using their software. They have an automatic pipeline called RootForce, and we were able to demonstrate that this worked with some tweaking. We were able to get this to work quite well in processing our own data. And now they've developed their own series of algorithms, some of those mimicking root system architecture modeling, and they also have other approaches. So if you look at that link there on the bottom, Monica just published a paper with 4D analysis of variation in root growth traits. And so now we can actually extract a whole series of features that describe variation in root architecture. including root length, density, diameter, crown diameter, maximum depth, lateral root emergent set depth, the number of roots, length, width, volume, etc. And the amazing thing about this system is that when we do reconstructions at 200 microns, we don't see the fine lateral root hairs, but when we do this at 100 microns, we do. So we can actually shift back and forth between those two models to get a sense of how much of this variation is actually attributed to the fine root hairs versus the more coarse root architecture. And we're now using these systems. We're just nicely, to be perfectly honest, in the last few months, we've got the system up and running. And so we're now able to use reliably up and running. We're able to routinely evaluate a number of different crop plants, including maize. In this case, we're looking at dynamic root growth, as well as genetic variation in root architecture, for example. So the Institute of Plant Sciences at Purdue is represented by more than 100 scientists from 5 different colleges at the university. Not very many of them would call themselves plant scientists. Maybe half of them approximately might call themselves plant scientists, and the rest of them are important collaborators. And so I've listed just a few of the faculty that are involved in different aspects of plant science phenotyping research in the last few slides, but there's a number of other very important collaborations ongoing to address other issues and questions with regards to multi-scale phenotyping. We have a whole group of engineers collaborating with plant scientists on new tools and modes of data collection for multimodal remote sensing. We have, if you're familiar with field-based phenotyping, or if you're familiar with field-based analysis of plants, in many cases, the under canopy phenotypes, the ones you can't see from the air, are more indicative of whether telling you a story about the crop than the above the top of the canopy. And so we have another group working on under canopy remote sensing, a group working on ear photometry and maize. We've got a whole new group working on digital forestry. So we have a team looking at either planted or native forest ecosystems using all of the same tools I just described to characterize, for doing digital inventories, for determining species mixes, etc. A lot of work is now being undertaken with regards to disease detection and prediction, predictive models for plant water and nutrient phenotypes. I think the single most important, I've heard this question come up a few times, and I actually just put this in because I was reflecting on it since this morning when we started this series of talks, is transfer learning, is what we call it. How do we take the lessons learned, the biology learned from controlled environments, and apply them in field environments, and what we know from field environments and apply them in controlled environments? And so we actually have several different teams here at Purdue, not a lot of people, but several different teams that have expertise in data analytics that are using either empirical approaches, machine learning approaches, or biophysical crop models to support transfer learning in the context of using remote sensing for high throughput phenotyping, either in controlled or in field environments. So as I said, I tried to highlight, well, there's a lot of people at Purdue working in this space. I tried to highlight the individual researchers on the earlier slides that were doing work. This group here represents the key members of the Institute for Plant Sciences. Dr. Young is the director for digital phenomics. Chris and Jason are support controlled environment and field environment phenotyping activities. Augusto just completed a postdoc here and is now working at Bayer, and Julie's been helping us with infrastructure development. And I just wanted to highlight them as key players in helping us develop for support and helping us develop this infrastructure. Much of what I've described in terms of investment has actually come from the university, from the Board of Trustees, and they're calling those the Purdue Moves initiatives. So we've just completed Plant Science 1.0, we're moving through Plant Science 2.0, and with support from the college, we're looking to add additional capacity in high throughput phenotyping. And we have a whole series of both federal support and private sector supporters that have been helping us maintain the various research activities that I described to you. With that, I think we're getting close to being back on time. I didn't get us all the way back to on time, but it got us a little bit closer. So I appreciate, like I said, the chance to speak with all of you.
Mitch, thank you very much. For this great overview and insights into your facilities setup and the various research questions that are addressed. So the question is, or the talk is open for questions this way around. Mark has a question.
Hi, Mitch. Thank you very much for this nice talk. I was wondering about the X-ray computer tomography. So the signals were very, very clean. So how do you prevent scattering of these X-rays? Do you need extensive pre-processing of soil, or can you just use commercial soil and the system is precise enough to correct for that?
So the plant science group actually went through and they tested a lot of different soil mixtures. They're actually using a commercial soil mixture that works well in terms of root imaging and being able to get segmentation, clean segmentation. The other aspect of that is making sure that the pots are suitably dry. They can't be wet or water soaked or waterlogged and get any kind of good segmentation, because the water soaked soil looks a lot like a root. And so the group has actually been working with Fraunhofer and have been slowly, I think we're almost 2 years now since the system was installed. And it's taken us about 2 years, a year and a half, to really learn how to collect clean data. And honestly, it's been since probably March of this last spring that we've had a really reliable system that we know how fast we can call plants out of the system. We can queue them up. The analysis, it's not fast, but it's become relatively more routine.
Looks very good.
Also got a question to that one directly. Is it true, or did I got it right, that you have 4 plants per hour processed through that system?
Yes, that's 4 plants when we're scanning 400 millimeters. So we can scan at 200 millimeter height pots or 400 millimeter height pots, and at 400 millimeters, it takes 15 minutes to finish a scan.
Okay. Hendrik.
Maybe to follow up on the X-ray tomography, one of the nice things is that you can follow the roots over time. But have you found any negative effects of the scanning by X-rays of the plants?
I just asked that question myself last week. We had a group on campus, and I don't know all of the, what's a lethal dose. And one of the engineers who specializes in this space got up and said that the dose of X-ray radiation that you get from a single scan is, I think, 2 orders of magnitude below a level at which they would harm the roots. Because the question that came up was, we're interested in looking at both root development under semi-controlled good conditions, but we're also introducing, for example, pests, corn rootworms. We're looking at other species that are being evaluated. Right now, we're developing algorithms to characterize rhizobial interactions with legumes, and how fast does that happen and how quickly can those be detected. And in our last 2 talks ago, I guess we were talking about using high throughput phenotyping for analyzing GM and CRISPR types of plant events. And so there's a lot of interest in a couple of our basic science groups in trying to use this non-destructive technique to describe the plant pathogen interactions that are possible either with favorable symbiotic organisms as well as unfavorable pathogenic organisms.
But you never have tested plants that were either measured by X-ray or were not measured to see how they were different after 6 or 8 weeks or so?
It would be a good idea. We haven't done that. I guess I was listening too carefully to the engineers. They said, don't worry, it won't be a problem, but it's probably worth testing.
We tested it in Jülich, and there was quite some effect, I heard from my colleagues. So there is reason, not the first time or the second time, but if you go to repeat it 4 or 5 times,
I see.
you will. Yeah.
No, that's a good point. Thank you for the suggestion.
Okay, so we have also the system, in PSI, and it's exactly about the radiation. It depends on the exposure. So if they do 4, samples per hour, if they do the tomography. So it's about the radiation, just simply. So we did this experiment, and sometimes we do just like 2D, so which takes like 2 minutes, to get the one scan, and it may be faster. But if you want to have a 3D model, yeah, it's true. You need time, so you need like minimum 10 minutes, to get the one picture, and then it's about the radiation. But the general problem for us was always the water. So what he said is exactly, and then the problem is, to do this experiment if you have completely dry soil, because you affect that. So the water, the balance. At the moment we do, quite good experiment with the potatoes, because the object is bigger. Our resolution is 50 micrometers, and yeah, it's a pain, if you have wet, soil, and then you have the tiny roots. So it's quite difficult. But if it's dry, yeah, that works, because there's the contrast, the scattering of the water.
All right. One more comment by Philip.
Yeah, sorry. Yeah, just commenting on this, there might also be differences in terms of the species or living forms you're looking at. For example, while fungi tend to accumulate ionizing radiation, other plants may be not having, or not to such an extent, this kind of trait. So if you're specifically looking at mycorrhiza infection, maybe it makes sense looking into the accumulation over repeated measurements and exposure to ionizing radiation.
No, these are all good suggestions. I think one of the things that happens is when you have 600 plants in the growth chamber, we're not even looking at the plants weekly. We have stages of development. We're trying to characterize variations in root architecture in order to process the plants in a timely manner. And so many times the plants are only being scanned probably 2 to 3 times at the most, and sometimes only once. And so maybe part of my lack of concern was the relatively few scans each of these plants were getting, but I do appreciate the suggestion that we take a look at treated and untreated plants.
All right. Is there a further question here in the audience? I don't see any. Is there any in the chat? Yes, there's one by Rick. Fascinating. A second X-ray question. How long does one measurement take in the CT system with the 75 micrometer setting compared to the 200 micrometer setting?
So the 15 minutes is at 100 microns when we're set at 100 microns. So the two settings that we've been looking at relatively routinely is either scanning at 200 or 100, and the 100 micron setting is at it takes 15 minutes to do a 400 millimeter pot.
Right. Thanks a lot. So right now I don't see any further question. And thank you very much for this very nice presentation and the intense discussion. And I think we can then move towards closing the session of today.